Thank Connie for coming in. For those of us that do basic science and are in a lab what feels like 24/7 and we don't actually get out into the clinics, it's very humbling for us, and I know for students hopefully, to realize what we're doing and why we're doing it. And sometimes I think it's very easy when you're in a lab all day long and you don't see patients to sort of forget the impact of the sort of pathogens and viruses that we work with. So thank you again, and it is very, very humbling from a basic research point of view to hear your story. So what I thought I would do is, instead of telling you how West Nile causes these symptoms or what we know about the immune response to West Nile, to talk a bit more about the genetic studies that Mark alluded to, because really West Nile is not that different from any other virus. It induces the immune response the same as most viruses do. It responds to the immune response the same as most viruses do, and there's really not a lot about West Nile that is unique to any other viruses, in both how it induces symptoms and how it responds potentially to the immune system. So because here at Mac we did a lot of work trying to figure out what makes this different between fever and your invasive disease, because that's so poorly understood that there must be some sort of a genetic element. That's really the focus of what we were trying to do with this big NIH study. So I'm going to talk a little bit about that instead, and sort of the lessons that we've learned along the way, some of the frustrations that we learned, and why, even though you can pour a lot of money into a project, why sometimes research can be inherently very slow, or at least much slower than what we'd like it to be. So as I mentioned, this virus is no different from any other virus. Very, very similar. It induces a protein very rapidly, which is very important for viral infections, called type 1 interferon. This is just sort of a prototypic pathway. West Nile, as I mentioned, is not a lot different from any other virus. When it gets into a cell and starts to replicate, there are a number of different cellular proteins that respond to the viral infection, can recognize components of the virus, and induce this protein called interferon. Now, even though interferon doesn't work as well as people would hope as a therapy, it's probably, at least very, very early in infection, one of the most important molecules that your body can make against infection. And what interferon does is it binds to receptors on either the same cell or surrounding cells, and it sets up an antiviral program where it induces a whole number of different genes and proteins that collectively try to either prevent this cell from becoming infected in the first place, or from limiting the infection and limiting the spread of the virus. And so oftentimes, if you already have a full-on viral infection, interferon treatment is sometimes too late. And so that's why it really is a race between how much of a foothold can the virus get versus how quickly can you treat with interferon. Why for a lot of viral infections, interferon doesn't seem to have the sort of efficacy we hope, because the virus has already established its infection. And the whole point of interferon is try to prevent the infection from occurring in the first place. So as I mentioned, West Nile is no different from any other virus. It makes double-stranded RNA. It induces, it's been shown over the last several years to activate all of these molecules. So there's really nothing unique about this virus per se. But we do know from studies in our lab that the interferon pathway does matter. We used a subtype of West Nile called Kunjun virus. So almost all the data I'm going to show you has to do with Kunjun virus. And if we just look at replication of the virus, this is vero cells, which is a monkey kidney cell line, and then these are two human fibroblast lines. And as you can see, and this is a log scale, the virus replicates much, much better in vero cells. Well, one defining feature of vero cells is that they can't make interferon beta. They have a lesion in the locus that they can't make interferon. So this is a cell type that many of us use to grow viruses, because there is no interferon produced, and so these viruses can replicate logs better than in other cells. And in primary fibroblasts, they're very, very good at making interferon beta. In fact, interferon beta used to be called fibroblast interferon, and so we can see that interferon, at least at the early stages of the virus, does matter. And so when we look for genetic associations, you'll keep seeing the word interferon popping up more and more. So this just sets the stage. So how do we study? There's different ways that we looked at replication. We can actually titer the virus. If we want to get a little bit more specific, I'll just give you an example of one of the assays we developed in the lab. This is a single-stranded virus, so if you want to differentiate between just virus physically being there or the virus replicating, you can actually look at the different strands. You can look at the negative strand, which is indicative of the actual virus replicating. It's a positive-stranded virus. So if you see a positive strand, which we can set up the assay that we can differentiate between the two, you can tell whether the virus is just physically there versus whether it's actually starting to replicate or not. And so early on, you can see we see higher levels of the positive strand because that's just incoming genome. Whereas over time, you see an increase in the negative strand showing that it's actually replicating. So there are different ways to tell if the virus is just simply physically there or if it's actually replicating. So as Mark mentioned, the study that NIH funded was really to try to understand why do some people get neuroinvasive disease, which is very rare compared to just West Nile fever. And West Nile fever, even though it's called West Nile fever, the symptoms are the same for almost any virus that you get. So fever, lethargy, this is the symptoms that are caused when you make interferon and make cytokines, which almost all viruses will do. So we were really interested in what is it specific to those individuals that get the neuroinvasive disease, especially since it's not always individuals that are immunosuppressed or old or have comorbidities. We were really interested in the genetics. There must be, or we thought there must be some sort of genetic component. So I'm just going to quickly go through the 3 different genes that came up on the first cohort study and what we did to try to look at them and why these genes actually from the get-go were quite interesting. So the first one was RFC1, and this one had the highest, this was I think the only one that was actually statistically significant in our initial study, so we're very excited. It had a very good significance. As Mark mentioned, it's a DNA polymerase accessory protein. Even though this is an RNA virus, that was a little bit curious. It's a large complex. This particular gene product can exist with other complexes. It's known to regulate a transcription factor that's very important in production of cytokines. And cytokines are the soluble proteins that mediate a lot of the immune responses. It can interact with many other gene products. But most interesting to us, it was also identified as a protein that bound to the interferon-stimulated response element. So not a lot of work was done on this, but because now we have this connection with the interferon response, we became really interested in this particular protein. And because it had, in the initial cohort study, such a high association, we thought this was a great candidate. So the one thing that we first did was, okay, if it's important for the virus to replicate, then if we knock it out, maybe we'll see differences in replication. So we used siRNA, which is a technique to try to deplete the RNA and thus the protein in a cell. So this just shows an optimization. These are all controls. And here we have specific siRNA against the gene and the RNA for our FC1. And as you can see, if we look at the mRNA, it was really effective in actually depleting all of the RNA in the cell and thus depleting the protein. So now we have cells where we do have this protein, we don't have the protein. And what we were really looking for was the ability of the virus to replicate. So in all of our studies, we wanted to see, is this linked to viral replication or not? And as you can see, it's not. So whether we have RFC1 expressed or not, the virus replicates just fine. So even though all of these SNPs are just single nucleotide polymorphisms, we thought the worst case scenario, usually that doesn't affect if a protein is made or not. It usually has subtle changes. So we were looking at the drastic changes. Is this protein involved at all? So even doing something as drastic as depleting the protein didn't seem to affect replication at all. So because it was also an interferon-stimulated response binding protein, we next asked, well, does it affect whether RFC1 is there or not, the production of these interferon-stimulated genes? Our prototypic interferon-stimulated gene that we look at in the lab is ISG56. When viruses infect cells or you treat cells with interferon, this gene is really rapidly upregulated. And you can see whether cells have RFC1 or don't have RFC1. In response to the viral infection, we see beautiful upregulation of this and other interferon-stimulated genes. So again, it doesn't seem to affect the replication of the virus or induction of interferon-stimulated genes. So that was disappointment number one. But again, data are what data are, and you are not supposed to get disappointed with data, because data are what data are. So next we moved on to the next highest gene on our list that had a SNP identified. And again, this was a really interesting, potentially biologically significant protein. So this is ANPEP, which is human aminopeptidase N. As Mark mentioned, it's already known as a receptor for at least one virus, and now it's been implicated in some other viruses. And so at the time, it wasn't abundantly clear what all of the receptors for West Nile virus were. And so if this was involved, that could either increase or decrease, depending on the mutation, the ability of a virus to bind and enter into the cells. This could potentially explain some of the differences. So we received from different collaborators in North America a number of different reagents to start studying this. Again, we use the same sort of strategy initially that we did with RFC1, is to try to deplete it from cells. And again, here are all our controls, and we had very nice depletion both at the RNA level and then subsequently over time at the protein level. So if we look at the protein, and we even looked over time course of infection to see if that would change. And what we found is that we could get really nice depletion. But again, even if we had really nice depletion, the virus could still get in and replicate whether ANPEP was there or not. So even if it is using this protein for entry into cells, it didn't seem to affect the ability of the virus to get in and replicate. So most of this work is done in primary fibroblasts. When a virus initially infects it goes through, the dermis, the underlying, there's fibroblast epithelial cells, Langerhans cells. We had, most of the fibroblasts we worked with were adult skin fibroblasts, so it was a nice system to work with. But we also wanted to look into neuronal cells to see if this protein is even, maybe it doesn't have an effect in fibroblast cells, but it's more in neuronal cells. So we have a control cell type 293T. This is another neuronal cell type that we looked at. And we had a number of different, we also had 3T3 cells that overexpress the protein. For some reason, from a protein level, we think from the RNA, it was just less abundant that we couldn't pick up the protein, but we could pick up the RNA. But even in the 3T3 system, when we overexpress the protein, again, if we look, it doesn't seem to matter. So whether we're in fibroblasts or neuronal cells, whether ANPEP is there or not doesn't affect the ability of the virus to get in and replicate. So that was disappointment number two. So we had 3 genes we were really interested in. And so the next one was SCN1A. And again, this was a very interesting, it was number 3 on our list. It had nice association. And again, biologically, it made a lot of sense. It's a voltage-gated sodium channel, at least it's one of the subunits. There have been many, many SNPs associated with this particular subunit of this sodium channel. And mutations or SNPs that are associated with this particular protein have sequela that's seen often with the neuroinvasive form of West Nile. So neurological disorders, and so it seemed very plausible scientifically why this would have shown up on the initial study. And it had also been shown with HSV1 that neuronal activity of these channels can impact on this particular virus. And this is a virus that infects neuronal cells, it goes latent in neuronal cells, and if an individual is severely immunosuppressed, as Mark mentioned, it can also cause encephalitis. So this is what the protein looks like. It's a huge protein, which made it very, very difficult to work with. And all of the little dots and circles that you see are SNPs or mutations that have been identified in this particular protein. And the one we were interested in was the one way down here in the red. So it's a very actively studied protein, and we're very excited about this. So because this is specific for neuronal cells, and it's very, very difficult to work with primary human neurons, these are terminally differentiated, they're hard to get, they're hard to work with. Again, we use these SKN-SH cells because these are really a nice model system for working with primary human neurons. If we could show that SCN1A was expressed in these cells and in human neurons, but not in control cells, so that's a good start. And so now we really wanted to look at function. So how do you start looking at function? Well, this is where we got into patch clamping and a drug called tetrodotoxin. So patch clamping, and we did this in collaboration with Colin Nurse in the biology department, is where you can take a single cell and you attach basically electrodes, and they have a system set up that you can actually look at currents, and so you can measure the current within a cell. And so when it's a voltage dependent channel, you can look at activity of this. And tetrodotoxin is a toxin that comes from the pufferfish. So you might have heard that in some countries, especially in Asia, eating raw pufferfish is a delicacy, but you have to really trust your chef, because if the chef does not prepare the fish properly, you can die from a neurotoxin, and this is a neurotoxin. And what happens is this neurotoxin actually destabilizes or inhibits these voltage-gated ion channels. So that's how it blocks the potentials, and so that's how it works. And so in collaboration with Colin Nurse, a postdoc in the lab looked at, with patch clamping of individual cells, in the absence of tetrodotoxin, this is the sort of signal that you get, so you can monitor the activity of these channels. And if you add the drug, you can see that it nicely inhibits this activity. So this was not an easy, but a nice model system. But again, when we start looking at increasing amounts of the inhibitor, it had no effect whatsoever on replication of the virus. Which again is suggestive of the fact that if you inhibit, I mean, we don't know exactly what that particular SNP would do to that particular protein, but if you globally, again by an overkill method, completely inhibit the channels, it didn't seem to affect the ability of the virus to replicate at all. So what did we learn from a lot of money, many, many people, and probably 4 or 5 years of work? Well, if you deplete or inhibit these 4 particular proteins, it really doesn't impact on the replication of West Nile virus at all. Now, there are some caveats with these studies. We were looking at, as again, the overkill strategy of, instead of looking at the particular SNP, because in a lot of cases that was very difficult to work with, we tried to express the proteins that just contain the SNP as opposed to the wild type protein. With the protein, you saw how large SCN1A is. That was basically technically impossible. So we did the complete inhibit or completely knock down. We didn't really look on other factors other than replication that could be causing an effect. One is cytokine production. As it was already mentioned, sometimes it's not the actual replication of the virus that causes the symptoms, it's the subsequent immunological sequela that can cause the infection. Now that is usually tied very nicely with replication of the virus. If the virus does not replicate well, you're not going to get an overactive immune response, and therefore you're not going to get the sequela. But in a lot of times, this is true for influenza virus, for example, it's not the actual virus that causes the issues, it's the overactive immune response. And that's really difficult even to test in vitro. We can look at cytokine production, but these studies really need to be done in the right animal model. West Nile, even though you can do work in mice, it's not a mouse virus. So again, there's not a lot of great model systems, and it's not easy to do those studies. People do them, but again, there are a lot of caveats with those sort of studies. However, these findings ended up being consistent with the one replication study that was done, where when a second whole cohort of fever and neuroinvasive patients were studied, these 3 genes didn't come up as being significant anymore. So, but of course it took those 5 years that we were doing all the work to get all of the cases and all of the samples and do all of the genotyping. So again, this is one of the realities of doing these sort of studies. And you might see in the literature, as I'll show you in a minute, there are a lot of gene association studies, there are a lot of GWAS studies, SNP analysis. The validation is very, very difficult. So even though we had 3 genes that biologically made a lot of sense, the fact that they didn't seem to be involved was actually validated when those particular SNPs did not come up significant in the validation study. So it's an inherent problem with these sort of experiments, but until you do the experiment, you're not going to know, right? So science is what science is, and data are what data are. So what we do know, though, it's really unknown whether host genetics actually play a role, although it's very likely that it does. But again, it's almost like looking for a needle in a haystack, but I still think it's worth doing because there's not a lot of other basic biological science reasons to really explain neuroinvasive versus West Nile fever. So that begs the question, does genetics really play a role even in West Nile virus infection at all? Well, the answer seems to be yes, again, with the caveats that a lot of these studies need to be replicated. So recently, well, between 2005 and now, there have been 2 papers that have implicated this pathway, the OAS, the oligoadenylate synthetase and RNase L pathway. So these are 2 different studies that have come out, and this is looking in humans. And just to bring it back to this interferon pathway that I was talking about, one of the classic genes that's upregulated by type 1 interferon is OAS. So again, this is a protein found within this whole interferon pathway. And so how OAS works is, again, interferon will induce the production of OAS. There is some OAS in cells, but interferon rapidly and very potently increases the levels. But what happens is when a virus comes in and replicates, almost all viruses, and again, West Nile is no different than any other RNA virus for that matter. You see production of double-stranded RNA, viral RNA. This viral RNA activates OAS, which is found within the cytoplasm and in an active form. When it becomes activated, it forms a tetramer, and it forms these oligoadenylates. So multiple lengths of basically A residues. These oligoadenylates then activate RNase L, which again is inactive until activated by these oligoadenylates. And now you have, so you're inactive, and now you have an active RNase L. And this is an RNase, and so what RNase L will do is it will actually cleave the viral RNA into small pieces. So now the RNA cannot be used to make protein. So it's a way of preventing viral protein from being induced. So what had been shown before, well, it's known that the human OAS family has 3 different functional genes and also an OAS-like or an OAS-L gene that does not appear to be enzymatically active. So it's not abundantly clear what this particular protein does. But what had been shown is that some inbred mice were known to be really susceptible to West Nile virus infection. And so when they looked at the susceptibility locus to try to figure out why these mice are really susceptible to this particular virus, what they found was they linked it to the OAS1B gene. And so it had already been implicated that, at least in mice, that OAS is important in controlling West Nile infection, at least in mice. So in the first initial paper, they had 33 individuals that had West Nile virus, either fever or neuroinvasive. So now they're not trying to differentiate between fever or neuroinvasive. They're just looking at those with West Nile and those without West Nile. And they found 23 SNPs either in any of the OAS genes or RNase L. There was 1 synonymous SNP in OAS-L that occurred at a higher frequency. So this is the one that had any sort of statistical significance. And it was thought that that the SNP was near a splice enhancer, so you'd get an alternatively spliced product, and that this might have dominant negative effects. This was hypothesized, it wasn't proven, and again, because this particular enzyme doesn't have activity, it's not quite sure what its normal function is, but it thought it might be acting as a dominant negative. In the 2009 PLOS Pathogens paper, what they did is they looked at SNPs specifically within OAS1. This is the homolog to the murine protein, or to the murine gene, so they were interested specifically in OAS1. And they focused on this one particular SNP because it also is within the last nucleotides in an intron, and it acts as a splice acceptor site, so again could cause splicing. And so what they did find empirically in the lab is that if it had a G allele, this is optimal for splicing, and so the particular protein has very high enzymatic activity, whereas if it had an A allele, it prevented splicing, so the resulting protein, that configuration, actually had lower enzymatic activity. So now you have two potential proteins, one with activity and one without activity. And what they were able to show is that as you increase the number of A alleles, so if you have two, you have a higher probability of getting infected by West Nile, or at least showing symptoms of infection, whether it's fever, neuroinvasive, but any sort of symptoms, than if you had both G alleles. And this is just the odds ratio. So again, if you were the genotype AA as opposed to GG, your chance of getting any sort of infection and subsequent sequela increased. And this actually, what they then did was they took human lymphoid tissue, cultured it ex vivo, they knew the genotype of the individuals, and they could show that over time, those with the AA actually always had higher levels of replicating virus. So they were able to validate that with this particular genetic lesion, and good evidence, not in these studies, but in other studies, that it's the activity of the enzyme that this results in the ability of the virus to better replicate. So that's a fairly good study that, again, makes biological sense. So the last paper, there's really only been 3 studies that I could find, at least in humans. The last one came out more recently in 2011, and what they did is they looked at 86 different candidate genes that are involved in the immune response. And what they found significant association with SNPs in IRF3, MX1, and again this OAS1. So OAS1 really does seem to be playing a role. It's been validated in a number of studies. And just to quickly show you where IRF3 sits, so IRF3 is an important transcription factor in making interferon beta, and MX1 is another protein that's produced by interferon. So there are a lot of nice studies, predominantly from Michael Diamond's group, using mice that are deficient in IRF3, showing that yes, IRF3 is important for the replication of West Nile virus, at least in mice. This has not been validated beyond that system, and I don't believe that the genetic analysis has been validated. And so far, there's no direct studies looking at West Nile and MX1, although this particular protein has been shown to have antiviral activity against orthomyxoviruses such as influenza virus. So it's still unknown, all of the genetic elements that play a role in either getting the infection, or if you have the infection, what sort of disease, whether it's asymptomatic, fever, or neuroinvasive. It's likely that components of the type 1 interferon they have been implicated and probably play an important role, because again, interferon is one of the very first cytokines that's made, and so normally what happens is, for most viruses, it's those really early events where you're talking about small numbers of virus particles, how efficiently it can induce interferon versus protect itself against interferon. That's probably going to determine whether you have an infection or you have clearance of the virus. So it's not surprising that all of the SNPs that have been, at least either experimentally and or through genetic studies validated, fall within this pathway. Although it is likely that multiple factors other than these will have an effect. And so finally, to acknowledge, there are a lot of people in my lab that worked on this very hard for 4 or 5 years in collaboration with Mark's group, and Jonathan Bramson and his research group were also involved in the NIH study. Thanks.